Welcome to the Deep Dive, where we cut through the noise and get straight to the most impactful insights from the latest research. Today we're exploring something incredibly fundamental. It's about how you and I perceive the world, something your brain does just effortlessly every waking moment, but which machines?
No.
Well, until now they found it incredibly challenging. Just think for a moment about your visual experience. Whether you're navigating a busy street, watching a game, or just walking in a park, your eyes and brain are constantly just bombarded with this overwhelming amount of visual information. We're talking something like 100 megabits per second of spatial and temporal input coming into each eye.
Each eye, yeah. It's a staggering figure.
Right. If you tried to consciously process every single pixel, every tiny shift in light, every movement all at once,
Yeah.
you'd just be completely overwhelmed,
Yeah.
paralyzed by the sheer volume. But you don't do that. Your brain has this really elegant subconscious solution. Selective attention. It acts like a natural filter, letting you automatically zero in on only the most important or salient regions of what you see. It just intelligently filters out all the irrelevant noise.
Yeah.
Okay, so let's unpack this. Our deep dive today is into some really groundbreaking research that truly bridges biology and cutting-edge engineering. The goal, well, it's to replicate this innate human ability in machines. We're focusing on a specific paper today. It's titled A Neuromorphic Proto-Object-Based Dynamic Visual Saliency Model with a Hybrid FPGA Implementation by an incredible team. Jamal Lariemolen, Chetan Singh Thakur, Ernst Niebuhr, and Ralph Etienne Cummings from top places like Johns Hopkins University and the Indian Institute of Science.
Really leading researchers in the field.
Absolutely. So our mission here is to understand how a model, one that's deeply inspired by the human brain, can not only accurately predict what catches our eye, but also process it incredibly fast, real-time speeds. And that opens up some genuinely revolutionary possibilities for the real world.
Yeah, and this isn't just, an academic exercise. It has really profound practical implications. Being able to understand and replicate visual saliency. It's critical for a huge range of applications that are literally shaping our future.
Like what, for instance?
Well, imagine an autonomous vehicle trying to navigate a complex intersection, right? If it can't quickly pick out a pedestrian stepping off the curb from, say, the complex pattern of a building facade, it just won't function safely. It needs that selective focus.
Okay, yeah, makes sense.
And the same idea applies to improving robotics. You need a machine to focus its attention on the relevant parts of a task it's doing. Or even in advanced surgical assistance systems, guiding a robot's gaze, maybe, to the most critical areas during a procedure and doing it in real time.
Right, you need that instant focus.
Exactly. And what makes this particular paper so exciting, I think, is this dual breakthrough. First, they've developed this highly accurate neuromorphic model. That means it's built to mimic how our brains actually work. And crucially, this is a big one, it requires no training data.
No training data, that's huge.
It's a massive departure from traditional machine learning, and it offers some really significant advantages. And second, they didn't just create a theoretical model on paper, they've actually implemented it using a hybrid FPGA approach, which makes it incredibly fast.
FPGA. It's field programmable gate array, right?
Yeah.
Specialized hardware.
Precisely. Specialized hardware designed for parallel processing. So it's this powerful combination. The biological fidelity, how closely it mirrors our own biology, and that real-time processing capability. That's what really elevates this research, makes it so impactful.
Okay, so let's start by digging into the brain's secret weapon then. This selective attention and how it leads to what we call saliency. We mentioned that colossal figure earlier. 100 megabits of visual input per eye every single second. It's a truly staggering amount of raw data.
It really is hard to conceptualize.
Yeah, I mean, picture trying to stream dozens of HD movies simultaneously and trying to make sense of what's happening in all of them at the same time. As you pointed out, no human brain, and certainly not most engineered systems we have today, could possibly process all of that information in parallel, in real time, if it tried to give equal weight to every single detail. Just grind to a halt.
Completely overloaded, exactly. The human visual system, the HVS, it's evolved to cleverly bypass this huge computational bottleneck. Instead of processing everything with the same intensity, it uses selective attention, preferentially focusing on the most salient regions.
The important bits.
The important bits, yeah. Decades of research, psychophysical, neurophysiological, have piled up compelling evidence that our brain constructs this kind of internal saliency map.
Okay, like a heat map of what's interesting.
Kind of, yeah. Think of it as this dynamic internal highlight reel. It pinpoints the areas most likely to grab our attention almost automatically, and this internal map then intelligently guides our gaze and, critically, our higher level thinking, making sure we focus our, precious mental resources only where they're most needed at that moment.
What's really fascinating here is how this elegant biological insight translates so directly to overcoming some really big challenges in computer vision. We're not just trying to build machines that passively see pixels anymore.
Right.
We're trying to build ones that understand what's important within their visual field, just like we subconsciously do.
And that understanding is precisely why saliency acts as such a vital initial pre-processing step for pretty much any real-time, high-level visual task in engineered systems. Without it, computers would just be drowning in this unmanageable flood of data.
Right.
Saliency provides what we often call data triaging. It intelligently detects and then transmits only the most relevant information to the next layers of processing. This frees up huge amounts of computational resources for more complex tasks like object recognition or planning. It significantly reduces the sheer volume of data that needs handling, and crucially, it cuts down processing latency.
That delay between seeing and reacting.
Exactly, that delay. And you can see how vital this is across so many areas. Navigation and localization, for self-driving cars or drones, robust object detection, classification, tracking in busy environments, efficient image and video compression, smarter surveillance systems that flag genuinely important events, not just shadows moving, and accurate action recognition. In every single one, identifying what's salient first makes the whole system way more efficient and effective.
Now, when we talk about visual attention, it's really important, I think, to differentiate between two main components. There's top-down and bottom-up attention.
Crucial distinction.
Top-down attention is driven by our internal state, right? Our goals, our biases. Like, if you're actively searching for your friend wearing a red hat in a crowd, your brain is using those top-down cues, the goal, the memory of red hat, to guide your attention. But this particular study, and saliency in the way we're discussing it today, focuses squarely on bottom-up attention.
That's right. Bottom-up saliency is purely a function of the inherent properties of the visual stimulus itself. It's that abrupt flash of light in your periphery, or the sudden movement, or maybe a strikingly different color that just grabs your attention before you even consciously decide to look.
But what was that moment?
Exactly. That pre-attentive, stimulus-driven response. It's automatic, and that's precisely what the authors are modeling in this paper.
And historically, some of the early, really influential models of saliency, I'm thinking of people like Koch and Ullman, or Itti, Koch, and Niebuhr. They were mostly feature-based.
Yes, that was the initial approach.
They focused on elementary visual features, things like color, orientation, intensity, contrast. They'd create these separate maps for each basic feature, sort of combine them, and then say, okay, this spot is salient because it's, say, bright red and vertically oriented, making it stand out.
Yeah.
It was a really powerful start, but, well, here's where it gets really interesting, because the field has evolved quite a bit since then.
Indeed it has. Newer object-based saliency models represent a really significant paradigm shift, and they find strong support from Gestalt psychology. That whole idea that the whole is greater than the sum of its parts.
Right.
Instead of just looking at isolated features, these models propose that our visual scenes are initially represented and manipulated in terms of these perceptual units, or what they call proto-objects. And these can be both static and dynamic.
Proto-objects. So not fully formed objects yet.
Kind of. Think of Renzink's coherence theory. It suggests that those elementary features we just mentioned, color, orientation, etc., they get rapidly and pre-attentively organized into these low-level proto-objects all across your visual field. They're like fleeting, temporary groupings.
Okay.
Initially, these proto-objects have pretty limited coherence, both in space and over time. They're kind of unstable. It's only when our focused attention lands on one of them that it stabilizes, and we perceive it as a fully coherent, distinct object.
Ah, I see. Attention solidifies it.
Exactly. And once you release that attention, it kind of dissolves back into its more dynamic proto-object state. These are really powerful ideas, and they were actually foreshadowed by earlier work, like Treisman's feature integration theory, which is also talked about an early pre-attentive stage of feature processing before full object perception. This paper really takes that framework and formalizes how these dynamic proto-objects are identified.
So the authors of this paper make this crucial neuromorphic leap. They develop a proto-object-based bottom-up computational saliency model. It's built on the intrinsic properties of the visual stimuli and, really importantly, the very early neuronal dynamics of our own visual system. And a key advancement here, one that really makes it stand out from many previous models, is that this one explicitly integrates motion. Motion is just.
Mm.
It's a massive signal for saliency, both in biology,
Yeah.
how quickly you notice something moving out of the corner of your eye.
Absolutely critical.
And consequently, it's critical in this model, too. They're building on some prior work, sure, but they're doing it with a far more accurate and biologically plausible method for incorporating these vital temporal time-based features.
And this leads us right to their first major contribution, this
Yeah.
truly neuromorphic, dynamic visual saliency model. What immediately jumps out, for me anyway, is how it handles motion in a way that so closely mirrors biological processes. It feels
Wow.
very grounded.
Mm.
Furthermore, its operation is based solely on these neurophysiological properties. What that means is its parameters are fixed. And critically, no training is required.
That's the point you made earlier. Still sounds amazing.
It is. It's a huge advantage compared to conventional machine learning models. Those models, they demand enormous data sets for training, which is computationally expensive, takes a long time.
And can be brittle, right?
Exactly. They often struggle with issues like overfitting to specific data. They might fail to generalize when they see something new, and they're vulnerable to these things called adversarial attacks, basically, clever ways to trick the system. This neuromorphic model just elegantly sidesteps all those problems because it operates on principles that are already sort of embedded in our biological understanding of vision.
That no training required aspect is it's such a big deal. It doesn't just make it efficient, it makes it incredibly robust, doesn't it? What do you think were the biggest hurdles in actually designing a model that could perform so well without that typical machine learning diet of data?
That's a great question, and it really highlights maybe the genius of taking this bio-inspired approach. The hurdle is exactly what makes it powerful.
Right.
You can't just rely on brute force pattern recognition from data, right?
Right.
Instead, you have to deeply understand the underlying biological mechanisms, how individual neurons or groups of neurons actually respond to specific stimuli in the real visual system. So the challenge lies in translating that neuroscientific knowledge, which is often really complex and subtle,
Yeah.
into precise mathematical and computational components, filters, grouping mechanisms.
Getting the math to match the biology.
Precisely. The authors had to meticulously design each filter, each grouping mechanism, so that it intrinsically responds to salient features and motion because of how human brains seem to be wired, rather than because it learned to from seeing millions of examples. And this makes the model inherently more generalizable and less susceptible to the quirks or biases of any specific data set.
That makes perfect sense. And the proof, as they say, is in the pudding. This model actually outperforms current state-of-the-art dynamic saliency models when it comes to predicting human eye fixations. That's the gold standard, right? The ground truth for saliency.
It really is.
So it's a truly bio-inspired powerhouse that actually delivers on its promise. Okay, now that we understand the why behind saliency, why it's so critical for both brains and machines, let's dive into the how. The author's model is called PODVS. That stands for Proto-Object-Based Dynamic Visual Saliency. And it's meticulously designed to mimic the early stages of human vision, specifically how our brains process visual input to rapidly identify what's important.
Yes, the core of PODVS lies in its use of what are called separable space-time filters. It's a really biologically elegant way to incorporate motion information. The model is directly inspired by specific types of simple cells found in two major pathways in the human visual system, the magnocellular and the parvocellular pathways.
Okay.
These cells are fascinating because they don't just extract spatial information, where things are pre-attentively, but critically, they also extract temporal information, how things are changing over time. So essentially, they're looking at both the location and the movement characteristics of things in the scene very early on.
Right.
And while PODVS cleverly builds upon an existing proto-object model that was designed for static scenes, its really critical innovation is adding these temporal characteristics. That's what allows it to powerfully process dynamic visual stuff like videos.
So to understand PODVS, maybe let's briefly touch on the foundation it builds on. That Russell et al. Static proto-object model for still images.
Good idea. Yeah. Imagine you take a standard RGB image, like a photograph, maybe 640 by 480 pixels. That static model would first deconstruct this image into several fundamental components.
The basic features we talked about.
Exactly. It looks at overall intensity or brightness, but also at color differences, think red versus green or blue versus yellow opponency. And it analyzes different orientations, horizontal lines, vertical lines, 45 degrees, 135 degrees, 9 feature channels in total for that static model.
Okay, 9 channels. Then what?
Then for each of these basic feature channels, the model essentially creates this multi-layered representation. It generates what's called an image pyramid. It's like having the same image information at several different scales or resolutions, coarser ones down to finer ones, downsampled across multiple levels.
Why do that?
That ensures the model can detect salient elements regardless of their size in the image, whether it's a small, distant object or a large, close one. Scale invariance, basically.
Got it.
Then, within each of these scaled layers, the model starts to compute what's called proto-object activity. This is where that magic of grouping begins, inspired by how our brains start organizing visual features. It involves these sophisticated operators that detect edges and identify areas that stand out from their immediate surroundings, center surround mechanisms.
Center surround, like spotting a bright dot on a dark background.
Precisely, or a dark dot on a bright background. And a key part of this is computing border ownership activity. This is a really fascinating neural phenomenon. Specific neurons in our visual cortex, area V2, literally seem to know which side of a boundary belongs to the object itself. It helps distinguish the figure from the ground.
Wow.
So this border ownership information is then integrated, kind of in a circular fashion, to start revealing where these nascent proto-objects are forming.
Then you mentioned normalization before.
Yes, a clever normalization step is applied next. It's designed to enhance maps that contain, say, a single strong proto-object, and maybe gently suppress maps where there are multiple, perhaps less distinct proto-objects competing, makes the strong ones pop.
Okay.
After this, these multi-layered representations within each feature channel intensity, color, orientation, they get collapsed down, consolidated into what are called conspicuity maps. You can think of these as summarized maps of standoutness for each individual feature type.
So a map for how conspicuous the colors are, one for orientation, etc.
Exactly. And then a second normalization step is applied across these conspicuity maps. This one sort of fine-tunes them, enhancing maps that have just a few strong peaks of saliency and maybe diminishing those that have many comparable, less distinct peaks. It helps focus on the really salient spots. Finally, these normalized conspicuity maps are intelligently combined, usually through linear summation, to produce the final saliency map for that static image. It's quite an intricate dance, of feature extraction, grouping, and aggregation, all carefully designed to highlight what would naturally grab human attention in a still picture.
Okay, that makes sense for a static image. Now, connecting this back to the bigger picture, the really crucial addition in PODVS, the thing that transforms it from processing photos to understanding videos, is its biologically plausible motion processing. The authors state it pretty unequivocally. Motion is the most significant contributor to visual saliency in dynamic stimuli for us humans. So without modeling motion accurately, any model of dynamic attention is going to be seriously incomplete.
Absolutely. Couldn't agree more. The model focuses specifically on how our visual cortex extracts motion pre-attentively, really early on. It mimics certain non-direction selective simple cells, particularly in the brain's primary visual cortex, V1, and it essentially emulates two distinct pathways we have in the human visual system.
The magnocellular and parvocellular you mentioned.
Exactly. First, there's the magnocellular pathway. Think of this as the brain's rapid alert system. It's almost like a dedicated motion detector. It contains what we call strongly phasic simple cells.
Phasic meaning responsive to change.
Responsive to change, yes. These cells have very high temporal resolution. They're incredibly good at detecting rapid changes over time. They have high contrast sensitivity, so they're great at spotting sharp light-dark transitions. But interestingly, they have relatively low color sensitivity. In the model, these are represented by a filter that has a strong excitatory phase, like an on response, followed quickly by a strong inhibitory phase and off response. This makes them incredibly sensitive to sudden movements or things appearing or disappearing, changes in brightness. It's like a quick, almost visceral reaction to dynamic events.
And the other pathway.
The second is the parvocellular pathway. This is more like the brain's color and detail specialist. It contains weakly phasic simple cells. These cells have lower contrast sensitivity but much higher color sensitivity and lower temporal resolution. They're slower to respond.
So kind of the opposite profile.
In some ways, yeah. They're modeled with less pronounced on-off phases. This makes them less sensitive to sudden motion compared to their magnocellular counterparts. Instead, they tend to provide more sustained responses, capturing the rich detail and color information from static or maybe slowly changing parts of a scene.
And the model uses specific math, specific functions to simulate these different temporal responses.
Yes, it uses a specific transfer function based on neurophysiological research to simulate these temporal receptive fields. What's really critical is that these biologically inspired filters, they strongly respond to the onset and offset of objects in the scene when something suddenly appears or disappears, but much less so to stimuli that are just sustained and unchanging.
Just like our eyes tend to ignore things that aren't changing.
Precisely. And this behavior emerges without any learning or prior training. It's hardwired into the model structure based on the biology. It's quite remarkable, really.
Okay, so that brings us to the actual processing steps.
Yeah.
How does it extract this spatiotemporal activity? Let's call it processing step P1.
Right, P1, extracting spatiotemporal activity. So the model gets a stream of RGB video frames, say standard video at 24 frames per second. It processes them strategically across its different channels. For the intensity channel, the brightness information, it applies that strongly phasic temporal filter.
The motion-sensitive one.
The motion-sensitive one, yeah. This involves a mathematical operation called convolution applied across the current frame and several previous grayscale frames. This is where it effectively captures that highly sensitive motion information, almost like a flicker or change detector.
Okay. And for color?
For the color channel, the model applies the weakly phasic temporal filter. This is done across the current and previous RGB frames. This approach helps retain more of the static regions of the scene and keeps that high color sensitivity that's characteristic of the parvocellular pathway. The outputs from this step are then used to create those 4 color opponency subchannels: red, green, blue, yellow, etc.
And orientation.
Interestingly, for the orientation channel, there's no temporal filtering applied here in this model. It simply uses the current grayscale frame. This preserves the static orientation information, which is another crucial aspect of saliency, but one that doesn't inherently rely on change over time in the same way.
Okay, so P1 extracts the raw spatiotemporal info. Then this flows into the full grouping mechanism, right? The sequence of steps, P2 to P7, that builds up the proto-object representation. This is where the features start to, well, group together.
Exactly. This is where the raw features start coalescing into more meaningful perceptual units. It begins with P2. Generate frame pyramid. Just like the static model, this achieves scale invariance. Those initial spatiotemporal responses are successively downsampled to create multiple layers, maybe 5 octaves or 10 levels, ensuring objects of various sizes can be detected.
Okay, same pyramid idea.
Then P3. Complex edge and center surround filtering. This is a big step. Here, it uses specialized filters called Gabor kernels, which are great at picking out edges and specific orientations, very much like complex cells in V1. Simultaneously, it uses differentive Gaussians, or dog kernels, to detect those center surround responses areas brighter than their surroundings when in center, or darker, OF center, similar to neurons in the retina and LGN. This lets the model find borders and strong contrasts effectively.
So finding edges and spots. And then it gets even more intricate, right? With the von Mises filtering and summation. That sounds complex.
It is quite intricate, yeah. P4 is von Mises filtering. This is where the complex cell responses, those edge detections, are modulated or refined by the center surround activities. The goal here is to compute those crucial border ownership responses we talked about, figuring out which side of an edge belongs to the object. It's computationally intensive, involves maybe 16 parallel filtering operations per pyramid level.
Wow, okay. Parallel processing is key there.
Absolutely. Then P5 is von Mises summation. This step integrates these border ownership responses across the different scales or levels of the pyramid, accumulating the results to get a more robust understanding of where the borders are and which way they face.
Building up confidence in the borders.
You can put it that way. This leads to P6, final border ownership responses. Here, the model sums up the activity for left-sided borders and right-sided borders, based on both the ON center and OFF center pathways. It consolidates all that border information.
Okay, so now we have a good idea of where object borders are. What's the final grouping step, P7?
P7 is grouping responses. This is where all that meticulously gathered border ownership activity gets integrated. It uses an annular or sort of ring-like integration method. It creates spatial masks based on the border ownership signals templates indicating which side a border belongs to. These masks are then convolved with specific kernels to finally compute the grouping responses. This is essentially where the dynamic proto-objects emerge, representing the figure-ground relationships in the moving scene. It's figuring out what's figure versus what's ground.
The culmination of all that filtering and integration.
Exactly. And then, to wrap it all up for the final saliency map generation, conspicuity maps are generated again for each subchannel: intensity, color, orientation. They get normalized, rescaled to a common size, and summed. Then there's that second, final normalization step across these combined conspicuity maps, similar to the idiot-all model, and they are linearly summed to form the instantaneous dynamic saliency map for the current video frame.
Frame by frame.
Frame by frame, this whole sequence repeats, culminating in that dynamic map, highlighting what our biological eyes would likely find most compelling in that moment of the video. It's a really detailed, multi-layered process, but one that genuinely mirrors the complexity and hopefully the efficiency of early biological vision.
All of this complex biological modeling. It's fantastic. A real achievement in computational neurobiology. But here's the rub, right? If a model, no matter how clever, can't run fast enough, it's just not going to be truly useful in those demanding real-world applications we keep talking about.
That's the crucial bottleneck.
Yeah. So it raises the important question. How do you take a computationally intensive model like PODVS with all those complex filtering steps, all those calculations, and make it run in real time? That grouping mechanism we just walked through, P2 to P7 especially,
Right.
that requires an immense number of these multiply accumulate operations, M&Es.
Billions of them, potentially, for a decent resolution.
Billions.
Right.
So for things like guiding a drone through trees or controlling a robot arm in surgery, real-time processing isn't just nice to have, it's absolutely essential. And standard software implementations running on typical CPUs, they just aren't fast enough for these kinds of critical, time-sensitive jobs.
No, they often struggle to keep up. And this is precisely where the author's really novel hybrid FPGA implementation, which they call PODVS FPGA, makes its entrance. They're using specialized hardware, an Opal Kelly XEM7350 board, which features a Kintex 7 FPGA.
Okay.
And the strategy is ingeniously hybrid. The most computationally demanding tasks, primarily those MSC-heavy operations deep inside the grouping mechanism, they get offloaded to the FPGA, which, as you said, is designed for massive parallel computation.
Well.
Meanwhile, the less demanding tasks, things like the initial spatiotemporal extraction, P1, and the final normalization steps, those run on a host CPU. In their setup, it was a pretty powerful Intel quad-core i7.
Splitting the work intelligently.
Exactly. The huge advantage of using an FPGA here is its ability to enable highly efficient pipelining and parallel processing. You can think of it like building the super efficient assembly line where lots of different parts of the calculation are happening simultaneously, not just one after another like on a typical CPU. This gives you a dramatic speed up.
Right.
And while this is a hybrid approach for now, using both CPU and FPGA, it's also a really significant pragmatic step towards their ultimate goal: a full end-to-end low-swap hardware solution.
Well, low size, weight, and power.
Exactly. Absolutely essential for embedded systems, things you find in drones, mobile robots, wearable tech, where resources like battery power and physical space are really constrained.
Okay, let's talk about how this hybrid system actually works in practice then. Communication between the FPGA and the host CPU.
Yeah.
That must be critical, right? Getting data back and forth quickly.
Absolutely critical. They use a high-speed USB 3.0 connection, which provides pretty good bandwidth, and they leverage Opal Kelly's front panel API for the software interface. They designed it carefully so that the communication latency, the time it takes for data to travel between the CPU and FPGA, is minimized. They found it was effectively insignificant within the overall processing time for the frame sizes they were using. So moving data wasn't the bottleneck, which can sometimes be a real challenge in these hybrid systems.
Good. And you mentioned they used a lower input resolution for the hardware version.
Yes, to manage the FPGA's resources efficiently, especially in this initial implementation, they used a lower resolution, like 112 by 84 pixels, compared to the full 640 by 480 software model. This allowed them to fit the complex parallel processing onto the specific Kintex 7 chip they have, while still proving the concept works effectively.
Okay. So how did they distribute the processing steps? You said P1, the spatiotemporal extraction, stayed on the host CPU.
That's right. P1 runs in software on the host. It mainly involves simply 1D convolutions, not a huge number of MAs, so software processing is efficient enough there. Adds maybe around 30 milliseconds of latency, which is manageable.
Then P2, generating the frame pyramid, that moves to the FPGA.
Correct. P2 shifts onto the FPGA. This step is crucial for scale and variance, and they use a clever nearest neighbor downsampling method. On an FPGA, this can be done extremely quickly using simple bit shift operations for calculating pixel addresses. Takes only a handful of clock cycles. Very efficient.
Okay. And then P3, the complex edge and center surround filtering. That sounds like prime FPGA territory.
Absolutely prime territory. This is where the FPGA really shines. It's a massively parallel operation. They run filtering calculations for multiple edge orientations and the ONO of F center surround kernels simultaneously across the image data. These weighted sums, the MSE operations, are exactly what FPGAs are built to accelerate. This really intensive step, which would bog down a CPU, is handled with impressive speed on the hardware.
Makes sense. And P4, the von Mises filtering for border ownership.
Yeah.
Also FPGA.
Yes, P4 also runs entirely on the FPGA. This involves numerous parallel filtering operations for each pyramid level, maybe 16 filters running at once per level. It's another highly parallelized MSC-intensive task that benefits hugely from the FPGA's architecture.
Okay. P5, von Mises summation.
Still on the FPGA, they use dedicated processing units, finite state machines, or FSMs, to sum up the responses efficiently without needing much extra memory.
And P6, the final border ownership consolidation.
Also efficiently handled on the FPGA. More parallel FSMs consolidate the border information.
Right. Now, P7, the grouping responses. You said this was a hybrid again?
Yes, P7 demonstrates the hybrid nature clearly. The initial generation of the spatial masks, which are needed to figure out which side of a border belongs to the object, that's actually done back on the host CPU. It's a less computationally demanding task. That mask data is then quickly sent over to the FPGA. And it's on the FPGA that the really heavy-duty calculation happens, computing the actual grouping responses. Those complex convolutions using the masks are performed in parallel on the FPGA, bringing that crucial figure ground understanding to life at high speed.
Got it. So mass generation on CPU, heavy convolution on FPGA.
Precisely. And then finally, the very last steps, the final normalization and the actual generation of the saliency map, those are processed back on the host CPU again. The grouping results are transferred back from the FPGA, and these final normalization and merging operations, while essential, aren't quite as MAC intensive as the core grouping, making them suitable for the CPU to handle.
Okay, that breakdown makes sense. You mentioned the current FPGA they used had resources for, say, one channel at a 112 by 84, or maybe 3 channels if they drop the resolution a bit.
That's right. It was a proof of concept based on the available resources on that specific Kintex 7 board. But the ideal scenario, the clear path forward for maximum speed, is to compute all 9 feature channels completely in parallel on a more powerful FPGA.
And that would give roughly a 9x speed up for the grouping part.
Potentially, yes. A near 9x speed up just for that computationally intensive grouping activity processing, which is really where the bottleneck lies. That would be truly remarkable for real-time performance.
Okay, so we have this sophisticated, brain-inspired model, PODVS.
Yeah.
And we have this clever hybrid hardware implementation, PODVS-FPGA. But the big questions remain, how well does it actually work? Does PODVS truly mimic human attention? And does the hardware deliver on its promise of speed without sacrificing accuracy?
Absolutely critical questions, and the authors tackled them with really rigorous validation. To check how well PODVS mimics human attention, they compared its predictions directly against actual human eye fixations on a set of videos. They use a standard, well-known dataset called CRCNS. It includes 50 videos, and crucially, it comes with precise eye fixation data recorded from 8 different human subjects watching those videos.
So you have a solid ground truth to compare against.
Exactly, a very reliable benchmark. They use 2 standard metrics for evaluating saliency models, AUC, ROC, and KLD. But importantly, they modified them slightly to address a common issue in saliency research called center bias.
Center bias. People just tend to look at the middle.
Pretty much. Many models, and even human observers sometimes, have this tendency to focus more on the center of an image or video, regardless of what's actually happening there. To ensure a fair comparison and remove this bias, the authors specifically focused only on the locations where humans actually fixated their gaze, and they normalized the scores against what are called human scores. Meaning they evaluated how well their model predicted fixations compared to how well one human's fixations predict another human's fixations on the same video. It's a really stringent test of performance.
Okay, so those metrics, AUC, ROC, area under the ROC curve, and KLD, Kullback-Leibler divergence. Can you briefly explain what they measure?
Sure. ACROC essentially treats the saliency map like a binary classifier. It asks, how well does the map predict whether a location was fixated by a human or not? A score of 1 means perfect prediction, while 0.5 is basically random chance. Higher is better.
Okay. And KLD?
KLD, Kullback-Leibler divergence, measures the difference or distance between two probability distributions. In this case, it compares the distribution of saliency values at the points where humans actually looked versus the distribution at random points. A higher KLD score indicates a better model. It means the model assigns higher saliency values to the places people actually find interesting compared to random locations.
Got it. Higher is better for both. So what were the results for the PODVS software model itself?
The results were quite compelling. They compared PODVS against 5 other leading bottom-up dynamic visual saliency models. Importantly, these were all models that also incorporate motion, don't require training, and had publicly available code, making it a fair comparison. PODVS significantly outperformed all 5 of these state-of-the-art models on the AUC ROC metric. It achieved the highest score among them. For KLD, PODVS also performed very strongly, matching or outperforming most of the others. There was just one model that showed a very slight edge, and the authors suggest this might be due to an additional texture feature used in that model, something that could potentially be integrated into PODVS in the future, too.
So top performance, especially on AUC ROC.
Yes. And what's particularly significant about this strong performance, even against well-established models, is that it provides really robust support for that Gestalt psychology idea and the hypothesis that attention is indeed object-based, not just feature-based. It suggests our brains really are looking for these cohesive proto-objects and how they change, not just isolated colors or edges when deciding where to focus.
Okay, so the pure software model is highly accurate, state-of-the-art even. Now let's turn to the hybrid PODVS FPGA implementation. How well did the hardware version maintain that accuracy? And crucially, how fast was it?
Right. For accuracy, they carefully compared the FPGA's output, which uses fixed point numbers, to the original software model's floating point numbers. There are always tiny inherent differences when you change numerical precision like that, but conceptually, the outputs matched remarkably well.
Okay.
When they tested the PODVS FPGA system on the CRCNS dataset, running at that lower 112 by 84 resolution, it still achieved very respectable AUC, ROC, and KLD scores. They were only slightly lower than the full software version. It's a small trade-off, mainly attributed to that reduced input resolution and the fixed point precision.
But they wanted to confirm its potential at full resolution, right?
Exactly. So, to definitively confirm its full resolution potential, they also ran a software emulation called PODVS Fixed. This version used fixed point precision, just like the FPGA would, but ran at the full 600 by 480 resolution in software. And this emulation yielded AUC, ROC, and KLD scores that virtually matched the original floating point software model.
So that proves the concept.
Yes, it effectively validates that a sufficiently powerful FPGA, one with enough resources to handle the full resolution and all channels in parallel, would perform comparably in accuracy to the original software model. And therefore, it would also outperform those other state-of-the-art models.
Okay, and on the resource side, you mentioned the limiting factors on their specific FPGA were things like DSP slices and block RAM?
Correct. Digital signal processing slices and block RAM. These are specialized memory and computation units on the FPGA chip. They provided detailed tables showing the resource utilization. And yes, processing all 9 channels in parallel, which is the ultimate goal for speed, would naturally require roughly 9 times these resources. That just points to the need for a higher-end, more capacious FPGA for a full, single-chip, high-performance solution.
Makes sense. But now, the speed. This is where it gets really exciting, right? The real-time breakthrough.
Absolutely. This is the game changer. Even with the limitation of processing the 9 channels sequentially, one after another, at that lower 112 by 84 resolution, the hybrid FPGA implementation achieved a frame rate of 2.08 hertz, or 2.08 frames per second.
Which might not sound super fast on its own.
Right, but compare it to the software version running on that powerful Intel quad-core i7 CPU. The FPGA version was already 2.64 times faster.
Okay, a decent speed up even sequentially.
But here's the truly mind-blowing result. When they calculated the performance achievable with an FPGA powerful enough to process all 9 channels fully in parallel, in that scenario, the PODVS FPGA implementation achieved a stunning 18.71 hertz, almost 19 frames per second.
Wow. compared to the software?
That's a 23.77 times speed up over the software implementation. And remember, this framework includes all the host CPU processing time as well. It clearly demonstrates the immense effectiveness of this hybrid approach in achieving genuine real-time processing capabilities for dynamic visual saliency.
That's the difference between a lab curiosity and something you can actually put into a robot or a drone.
Exactly. It pushes it firmly into the realm of practical, mission-critical applications.
This incredible speed really points directly towards that ultimate goal you mentioned, an end-to-end low-swap hardware solution. Low size, weight, and power. The authors highlight that those accumulate, the MAC operations, are the critical performance bottleneck.
They really are.
Processing a typical resolution frame, they estimate, requires billions of MPace. Implementing these efficiently on parallel FPGA hardware is absolutely the key to overcoming that massive computational hurdle.
And it's worth noting their deliberate choice of using an FPGA rather than, say, a traditional GPU, which people often think of for parallel processing.
Why FPGA over GPU here?
Well, while GPUs are certainly powerful, for applications that require a massive number of very specific operations like MX, and especially where power efficiency and a small physical footprint are paramount, FPGAs have actually been shown to significantly outperform even high-end GPUs. Research suggests FPGAs can offer better operations per second and, crucially, better operations per watt.
Better power efficiency.
Much better power efficiency, which is vital for those low-swap embedded systems. FPGAs excel at certain types of highly parallelizable tasks, making them often the superior choice when every watt of power and every cubic millimeter of space counts.
Okay.
So the ultimate goal really is that full end-to-end FPGA solution for real-time saliency computation, meeting those strict low-swap requirements, and the potential applications. They're just vast and incredibly impactful. Everything from improving image quality assessment and surveillance systems to enabling safer and more autonomous unmanned aerial vehicle navigation and obstacle avoidance.
You mentioned the drone flying through the forest.
Exactly. Imagine that drone autonomously navigating a dense forest canopy. It just can't afford to analyze every single leaf in detail. This kind of saliency model helps it instantly pinpoint the things that matter. The approaching branch, the moving animal, the gap in the trees it needs to fly through. It makes its flight path planning much safer and smarter, all happening in real time.
Yeah.
It could help ground robots explore complex, unknown environments more intelligently. And even guide advanced robot-assisted surgery, maybe by learning where a human surgeon typically looks and directing the robot's camera or highlighting critical areas automatically.
That's incredible.
And furthermore, the highly biofidelic nature of this model, its deep faithfulness to actual biological processes, makes it incredibly suitable for future implementation on emerging neuromorphic hardware. These are new kinds of chips designed to process information using spikes, much like biological neurons do. That could push the boundaries of low-swap computing even further, achieving truly brain-like efficiency.
Wow. Okay, what an incredible journey. From the fundamental way our brains handle overwhelming visual data, all the way to building specialized hardware that mimics that process in real time. Let's try to recap the dual breakthrough we've explored today.
Sounds good.
So first we have the neuromorphic, proto-object-based dynamic visual saliency model itself, PODVS. It fundamentally computes saliency much like the very early stages of the human brain, focusing on identifying these dynamic proto-objects that sort of pre-attentively exist in a scene. It's feed-forward, bottom-up, deeply rooted in biological plausibility. And crucially, as we've emphasized, it requires no training on massive data sets. That really sets it apart from typical data-hungry machine learning models. And the evidence shows it actually outperforms other state-of-the-art dynamic visual saliency models in predicting where human eyes will actually look in a video.
A major achievement on the modeling side. And then the second, equally vital breakthrough, it's novel hybrid FPGA implementation, PODVS FPGA. This is what truly unlocks the potential for real-time processing in real-world applications. We saw how this clever hybrid approach, strategically offloading the most computationally intensive parts to the FPGA, achieves that stunning 18.71 Hz processing rate, nearly 24 times faster than the software version. And it does this while meticulously maintaining high accuracy in its saliency predictions. This work brings us significantly closer to that goal of a full, end-to-end, low-swap hardware solution, really pushing the boundaries of how machines can perceive and interpret our dynamic world.
So what does this all mean for you, our listener? This deep dive, I think, truly highlights the incredible power of bioinspiration. By carefully dissecting and understanding the intricate workings of our own visual system, how we subconsciously triage this overwhelming flood of visual information in mere milliseconds, we can engineer remarkably powerful, efficient, and importantly, robust solutions for machines.
Yeah.
From enabling drones to more effectively dodge obstacles in real time, to potentially guiding the precise gaze of surgical robots. This research is just a profound testament to the potential of biomimicry, looking to nature for answers.
Indeed. And it really leaves us with a truly thought-provoking question to ponder, doesn't it? If just mimicking the human brain's initial pre-attentive visual processing leads to such massive leaps in machine efficiency and accuracy, what possibilities might open up when we can fully integrate these kinds of dynamic proto-object computations with higher-level top-down processes, things involving learning, memory, contextual understanding.
Yeah.
Imagine machines that don't just see what's salient, what stands out, but can truly understand its significance and the broader context of the situation, much like we intuitively do. This deep dive into neuromorphic engineering, it's actively pushing those boundaries, reshaping how machines might perceive and interact with our incredibly dynamic world in the future.